Hey, it's Corona time right now.
Coronavirus disease 2019, or COVID for short, needs no introduction. This deadly pandemic has swept through the world, invading virtually every facet of our lives. Individuals affected with COVID 19 experience a variety of symptoms, ranging widely from fevers, chills, loss of taste and smell, fatigue, shortness of breath, headaches, nausea, muscles and body aches, congestion, amongst a multitude of other symptoms. COVID 19 spreads through air droplets, making it crucial to take preventative measures to contain the spread of the virus, such as masking up, avoiding crowds, social distancing, and quarantining if exposed. Contact tracing is also a very useful method to try to gain understanding of the infection and transmission of the virus within a community. Although this virus has been the center of much controversy and has played no little part in many ongoing current events, it is indisputable that returning to the same world before COVID 19 is impossible. With research for the cure underway, the most effective way to contain the spread of COVID is to efficiently identify and quarantine individuals who have been infected. Thus, having a reliable and quick way to test for COVID is essential. This is where the polymerase chain reaction tests, or PCR tests, come into play. Currently, there are two main types of tests that identify COVID: a diagnostic test and an antibody test. An antibody test, or a serological test, is a test that is taken by procuring a sample of a patient's blood and identifying the presence of COVID antibodies. This test reveals if a subject has been infected by COVID in the past, but it does not reveal if the subject is currently affected. Therefore, the antibody test is not a reliable test to identify if one has COVID. There are two types of diagnostic tests: a molecular and antigen test. An antigen test is a nasal test that identifies specific proteins in a subject's body to determine if they have COVID or not. Finally, a molecular test, such as the PCR RT test, is a nasal test that identifies a genetic sequence common in the viral genome and amplifies it 1,000,000 to 1,000,000,000 fold, allowing for identification of the virus's presence. This makes it the most effective and reliable test method to determine if a subject is indeed infected. RT-PCR stands for real-time PCR, or real-time polymerase chain reaction. RT-PCR is sometimes referred to as molecular photocopying. That measures amplification actively occurring in real time.
Before we try to understand how the PCR test works, there's one distinction that must be made clear. PCR tests only work on DNA strands, while the COVID-19 virus's genome only consists of single-stranded RNA. To bypass this issue, another form of the PCR test must be used, called reverse transcriptase PCR, or RT-PCR. This distinction is important to make, as RT-PCR allows the conversion of the virus's RNA strand into complementary DNA, or cDNA. Now we can start to explore exactly what RT-PCR does. In short, the RT-PCR test amplifies the sample of medium taken from a subject, and if the subject is infected, identifies a sequence of nucleotides common only to the COVID-19 virus. To start, a nasopharyngeal swab is inserted into a subject's nose and rotated to collect a sample. This sample is placed in a viral transport medium to prevent contamination. From this, the single-stranded viral RNA will be extracted so as to isolate it from unwanted components in the original sample. To isolate the RNA, the sample is transferred to a microcentrifuge tube and a lysis buffer is added. This buffer's purpose is to denature the environment around any potential viruses. After the medium with the buffer is mixed in a pulsed vortex and incubated at a room temperature, the denaturing of the medium will cause the virus to lyse, or break apart, releasing its components into the medium in the tube. The next step is to isolate the RNA from the other contaminants. This is performed by filtering the medium through centrifugation in a spin column. The spin column is made up of a stationary phase that consists of a silica gel matrix. Only the RNA will bind to the silica gel matrix, while the other contaminants, such as proteins, will pass through the bottom of the tube. Thus, by centrifuging the tube under optimal salt and pH conditions, the only component that should be left in the solid phase is the RNA. However, some contaminants may not have fully passed through the solid phase. Thus, another centrifuging must be performed, this time with a wash buffer. The wash buffer will flush away any remaining contaminants out of the solid phase. Once the contaminants are discarded and the spin column is moved to a new and clean centrifuge, a elution buffer is added. When centrifuged with the elution buffer, the RNA will detach from the solid silica gel phase into the tube. With this isolated RNA, we can now begin the process of reverse transcriptase to create complementary DNA. Again, this process is necessary, as PCR is only effective when paired with DNA and will not amplify RNA. For this process, a master mix must be created with all the components necessary to have the RNA undergo reverse transcriptase and begin PCR. The master mix contains a buffer, reverse transcriptase enzymes, dNTPs, forward primers, reverse primers, a Tachman probe, and DNA polymerase. The goal of RT-PCR is to amplify or make more copies of the target sequence in the RDRP gene, the E gene, and N gene in the coronavirus disease. These genes code for RNA-dependent RNA polymerase, the envelope proteins, and the nucleocapsid proteins, respectively. These proteins make up the structure of the coronavirus. Choice of the target genes depend on the type of primers and the probe sequences used. The reverse transcriptase enzyme adds complementary dNTPs, or DNA nucleotides for short. They add it to an RNA primer that attaches to a strand of RNA. This process creates a DNA/RNA hybrid, which is an RNA strand binded to a complementary DNA strand. To separate the two strands, the medium is denatured. This initial denaturation is responsible for the deactivation of reverse transcriptase, as well as the activation of the Tachman DNA polymerase, which synthesizes a complementary DNA sequence on the now separate cDNA. PCR consists of a series of thermal cycles. Each cycle amplifies the cDNA present in the medium exponentially, allowing for billions of copies to be made in a short amount of time. Each PCR cycle consists of 3 stages: denaturing, annealing, and extension. In the denaturation phase, the reaction chamber is heated to 95 degrees Celsius and used for the denaturation of the double-stranded DNA template, which separates them. In the annealing phase, the reaction chamber temperature is lowered to 58 degrees Celsius, allowing for the forward and reverse primer to anneal or attach to its complementary sequence on a single-stranded DNA template. Annealing temperatures rely completely on the length and temperature of its primers. The extension phase includes the Tachman DNA polymerase synthesizing new DNA strands complementary to the DNA template strand by adding free nucleotides from the reaction mixture that are complementary to the template. Temperature of this step depends on the type of DNA polymerase used. It is important to note that the first cycle of PCR is performed to create the double-stranded DNA template from the original RNA. During the annealing step of PCR, a Tachman probe binds to its complementary sequence on a DNA strand. The importance of this probe is that it is what allows one to identify the presence of the gene sequence related to the COVID-19 virus in the medium. The Tachman probe consists of a fluorescent receptor attached to a 5' end of the probe and a quencher attached to the 3' end of the probe. The existence of this quencher on the Tachman probe suppresses the fluorescence given off by the reporter. However, when the DNA polymerase synthesizes a new strand in the extension step, the Tachman probe is removed along with the primers and is cleaved. This cleavage of the Tachman probe results in the separation of the quencher from the reporter. This brings about fluorescence in the medium. With every cycle of PCR, more and more dye molecules are released into the medium. The concentration of the dye can be measured electronically if the virus is present initially. If the virus is not present originally in the medium, the probes do not bind to the DNA strands, and no fluorescence is observed. However, if fluorescence is observed, it can be concluded that the subject is infected by COVID-19.
Conventional PCR is slightly different from RT-PCR since RT-PCR is a variant of PCR. In PCR, denaturation, annealing, and extension are the three steps, while in RT-PCR, reverse transcription is followed by PCR, denaturation, annealing, and extension. In PCR, DNA polymerase is used as the enzyme adding nucleotides, while in RT-PCR, reverse transcriptase and Taq DNA polymerase are used as enzymes. The PCR test has many advantages over other conventional tests. PCR tests are fast, inexpensive, accessible, and reliable. The results of the tests are available after only 24 to 48 hours. Someone is not left in the dark for weeks after being tested. The testing process is only inhibited by the time it takes to send the sample to and from a special lab where the test is performed. They're also minimally invasive, only requiring a quick insertion of a nasal swab. Throat swabs and saliva samples are also used in certain scenarios for sample collection. However, RT-PCR tests that are sampled from the deep nasal cavity are observed to have fewer false negatives as compared to the other methods of collecting samples. PCR tests are accessible as they can be conducted at a hospital or a clinic, like most molecular tests. Additionally, the RT-PCR for COVID can also be taken from a patient's car or at home at his or her convenience. Some locations where they are available are hospitals, drive-up facilities, doctor's offices, commercial labs, local pharmacies such as Walgreens or CVS, and independent self-sampling kits. The PCR test also determines if the subject is currently infected with the virus, not only if they had previously been infected, as other tests do. The PCR test is also highly specific and can be altered to amplify evolved versions of a virus. It is also highly efficient, as many samples can be amplified at once. With all of its convenience, the PCR test is not without its drawbacks. False positives can arise due to a variety of factors. Samples can get switched or mixed up. There can be software glitches in the computer system storing sample data. Mistakes can also be made when entering or communicating data from one software system to the next. Since PCR allows for the high degree of duplication of the targeted genetic segments by PCR tests, which depends on the number of cycles of duplications run, can multiply small concentrations of these contaminated viral segments by 1 billion times or more. Therefore, small amounts of contamination can produce false positive results that are difficult to distinguish from true positive results. PCR tests can also lead to false negatives. Particularly, nasopharyngeal swabs are not comfortable for most people, and so the patients can make it difficult to collect the sample carefully and in the correct area. Not reaching deep into the nasopharyngeal region can lead to lack of sample collected, and thus lead to false negatives since the viral RNA wasn't located. It's estimated that errors can occur about 8 to 10% of the time, which results in false negatives. PCR is a useful tool used for a broad range of applications. Molecular applications of PCR tests include DNA fingerprinting, classification of organisms, screening for mutations, detecting pathogens, discovery of drugs, genotyping, and prenatal diagnosis. Some specific examples of molecular applications include genetic testing, detection of cancerous cells, and detection of pathogens. PCR is also useful in forensics. Human identification by PCR is reliant on amplification of unique short tandem repeat sequences, STRs, in gDNA or genomic DNA to differentiate individuals to help solve crimes. PCR applications are used for sequencing for bioinformatics searches, genomic cloning, and integrated into the Human Genome Project. Finally, PCR is also applied to genetic engineering, which requires the use of PCR for sire-directed mutagenesis and gene expression studies. Namely, PCR is useful in agriculture by amplifying target genes for food pathogen detection, plant genotyping for breeding, and GMO testing. RT-PCR, or real-time polymerase chain reaction, also known as molecular photocopying, can be done on any bodily fluid, saliva or secretions in the throat. But typically, a nasopharyngeal swab is taken in a clinical setting or in a patient's car or home. A provider moves a long cotton swab into a patient's nostril, stopping at the nasopharyngeal area where the sinuses meet at your throat, because that is where most of the virus lives in large amounts, if the virus is present. The cotton swab is swirled for a short time, 10 seconds usually, which leads to a feeling of discomfort. Most patients report. PCR can also be done on a swab, just inside the nose and from saliva samples, but these methods would not be effective ways to gather samples because they are less sensitive to finding the virus, and therefore the nasopharyngeal swab is considered the ideal golden standard. The RNA of the sample is placed into a medium or a liquid solution and extracted. RNA from viral particles are bound to complementary cDNA sequences and then copied. The copies of DNA are made through repeated cycles, exponentially amplifying the viral genome in a significant quantity, reaching a defined threshold for a positive test. However, if the sample obtained from a patient does not have specific coronavirus RNA, then very little amplification occurs, and the test never reaches the threshold for positivity. Therefore, the person would test negative. The impact of the COVID-19 RT-PCR test cannot be considered insignificant. The application to create a viable method to test for such a deadly pandemic has a great impact on the security and well-being of the general public. The security this test provides, along with its variability and ability to deal with any mutations, allows the RT-PCR test to have a significant impact today and in the future. As always, remember, take care, stay safe, and social distance as we navigate these unprecedented times.